My name is Gail Seabold, and I'm a postdoctoral fellow in the laboratory of Dr. Veronica Alvarez at the National Institute on Alcohol Abuse and Alcoholism. This work was performed in collaboration with Dr. Kathleen Grant and Andrew Rao at the Oregon Primate Center and Dr. James Daunais at Wake Forest University, who provided the non-human primate tissue used in this study.
The overall goal of this procedure is to introduce fluorescent dyes into neurons and label brain sections from different species, such as rodents and primates, of any age for the purpose of studying the morphology of dendrites and dendritic spines. This is accomplished by first coating tungsten beads with a lipophilic dye such as dye-eye. The second step of the procedure is to line tubing with dye-eye coated beads and cut it into small pieces which serve as bullets for the Helios gene gun system. The third step of the procedure is to shoot dye-eye coated beads into brain slices using the gene gun. The final step of the procedure is optional, as DiOLISTICs can be combined with immunostaining to simultaneously localize other markers. Ultimately, results can be obtained that show sparse fluorescent labeling of neurons suitable for high-resolution imaging, such as confocal or two-photon microscopy, and the study of dendritic branching and dendritic spine morphology.
The main advantage of this technique over existing methods is that DiOLISTICs can be applied to brain sections from animals of all species, ages, and genotypes. It can be used in combination with immunostaining and produces sparse fluorescent labeling that's suitable for high-resolution microscopy.
Before making